Okay, so let's get some, let me give you an example of this. I'll probably have one example to actually go through. So this was a study that we did a long time ago, looking at just what's a normal variation gene expression in the lung, right? So we took a lot of different people, different ages, different parts of the lung, smokers, non-smokers, etc. And what we found is actually location, if you take biopsies at different location of the lung, it doesn't really have a major effect on expression. But obviously, whether you're current or if smoking history plays a huge role, which you'd expect, but also age. We get a lot of differences in the age here. So one subset, so we've got old lung. These are 6 individuals with an average age of about 70, and these young individuals, 7 of them, a typical age around 30 years old. Okay, when we do the statistics, I just simple t-test, a non-parametric permutation test at a p-level of less than 0.01, we end up with 452 probe sets or transcripts with an age effect in human lung. Okay, so I'm starting there. Okay, like I said, you can take, we can kind of segregate this based on the intrinsic properties of the genes itself. So the extracellular environment plays an important role in lung physiology. So I decided to take a look at the extracellular proteins in aged lung. And what you can see, this is awesome. They're all increasing. And we actually see that from a physiological level, that we actually see an accumulation of the extracellular material as people age. All of these are aging, and you can actually group these into different processes. So all these in green are involved in collagen processing. These genes in blue are involved in the structural components of the extracellular matrix, growth factor signaling, immune response. And there's one gene down here. The only one that doesn't increase is surfactant D, which actually shows a significant decrease, right? You got all this. Again, this is just, this is just using visually to space this out. And so actually it turns out surfactant D is deregulated in a variety of pulmonary disorders, right? Now we can even go farther. Let's go farther down the rabbit hole, right? So we've got all these genes. What actually, what induces them? The changes in expression got to come from somewhere. What's causing this change? And this is just good old-fashioned detective work. And what we find is that the majority of these genes are actually induced by either TGF-beta or IGF-1. Okay? So I thought this was cool. This was actually when I first got into the pulmonary department. I thought pulmonary, it was blood vessels. I was like, you guys work on lungs? That's cool. So having known nothing about lungs, I searched TGF-beta, IGF-1, and lung. And instead of coming up with age, what I came up with was this disease, IPF1, idiopathic pulmonary fibrosis, right? So this is a gene, this is a disease that is involved in uncontrolled scarring of the lung. And here's the deal, it typically or most often affects elderly individuals. So we got that connection of age. That's kind of cool, right? So I decided, well, we got this connection here. I'm sorry. But that these 2 growth factors that I identified in my normal human lung, many people in the field believe are causative or are involved in the pathogenesis of fibrotic lung disease. So I'm seeing an increase in my activity. So what I did was, I went and I actually tried to find all the microarray studies done on IPF, pulmonary fibrosis. And I came up with 2. There actually wasn't a lot out there. And they're actually done by the same guy, Naftali Kaminsky. So I decided I'm going to compare, I'm going to download these array studies, generate lists, and compare it to my own aging study. Because we're already seeing a connection there with the TGF-beta and IGF-1. Let's explore that a little further, right? So I did that. I compared. And these are all the genes that are commonly, that were common to both array studies, differentially expressed in both array studies. Here's aged lung in this column and IPF lung in this column. Or if we look at TGF-beta, we can see that, or this is TGF-beta induced. So this is a gene that's induced by this growth factor. And you can see the fold changes are almost the same. So it kind of argues against these growth factors being pathogenic in IPF, I would say. I would say that they might play a role, but they're not the sole cause. R1, we see a significant decrease in our normal aged individuals, and we see a huge increase, right? So I saw this, again, it's that pattern recognition. Find what's different, right? Bam! I thought, oh man, I'm going to cure this disease, blah, 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 fireworks are going off. I thought I was, on my way. And then I searched DDR1 and find that people have already found this connection, right? That this gene contributes to the survival of lung fibroblasts in IPF. And not only that, but mice that are deficient in this gene are resistant to bleomycin-induced fibrosis. But you can see, though, I mean, you're seeing the logical steps here, right? That we're, you're using the biological information. You're using these differences to lead you to conclusions. And this made me feel good because it validates the method that I was using, right? I see this difference here, and it's obviously important. And other people have seen it's important. So there was another study. So as I was doing my analysis, I found this. There's an array study done on old mice lungs, right? So they looked at gene expression changes in 2 strains of mice of different ages. So this is a shorter, Black 6 is a longer-lived mouse than the DBA. And so this would be the old DBA mice, this would be middle-aged Black 6, and this would be old Black 6. And so there's 18-month versus 2-month-old. And so I downloaded this array study. I got a list of genes with an age effect in the mice. And when I compare it to my study, my genes with an age effect in humans, I'm only getting like 45% in common, 4.5% in common. That's very low. I'm like, what's going on here? Not only that, but if you actually look at, concordance rates, if it goes up in humans, does it also go up in mice? We're only getting like 30% agreement. That's crazy. So I took that array and I did the same thing I did with IPF. I took the genes with an age effect in mice, compared it to my genes with an age effect in humans. And this is what I get for those common genes. That looks different. Why do you think it does? If we only had a pulmonologist in the audience. What do you think that is? What's causing that?
The difference
in the mice. Why does the old human lung look so much different than the old mouse lung?
There's different exposures and the length of there's longevity of a mouse versus longevity of humans is very different.
Right. I mean, you've got environment. I mean, the lung is, completely vulnerable to the environment, right? This is 70 years of exposure to a smog-related environment versus, a little over 2 years, right? So maybe this is the effect. So how could I test that? How could I think that, if I think of environment's causative, what could I compare to this to maybe say that might be true?
How about you expose your mice to smoke?
Exactly. Or bleomycin, which I was able to find. So that's what I did. I went out and I tried to I found an array study where they just took adult Black 6 mice and treated with bleomycin versus saline. And so these are actually very young mice, with a huge damaging effect. And now it's starting to look a little bit more like the human, isn't it? I think I'm going to stop there. Yeah, so.